Our speaker tonight is Dr. Fuz Rana, RTB's VP of Research and Apologetics. Fuz earned his PhD in biochemistry at the University of Ohio, and he did postdocs at the universities of Virginia and Georgia on cell membranes. He worked as a senior scientist in R&D at Procter & Gamble for 7 years before coming to RTB. He's published articles in peer-reviewed scientific journals and delivered numerous presentations at international scientific meetings. He's made countless media appearances, including on Fox News, The John Ankerberg Show, and Harvest Television. He's the author of many books, including Origins of Life, Who Was Adam?, Creating Life in the Lab, Humans 2.0, Building Bridges, Thinking About Evolution, and my personal favorite, The Cell's Design. He and his wife, Amy, live in Southern California, and they have 5 children and 10 grandchildren, and I understand another one on the way. So please join me in welcoming Fuz Rana. And at this point, I'm going to stop sharing and ask Fuz to share his screen.
Well, good evening, everybody. Thank you so much, Kirby, for inviting me to hang out with you guys tonight. And let me go ahead and share my screen here. Let's see if I can do this. All right. Is everybody seeing the screen okay?
Looks good.
All right, good, good. Well, anyway, Kirby asked if I would address the question tonight, can science provide evidence for faith? Can science provide a basis for faith? And so I'm going to take on that question. But before I do, I just want to make sure that I take a moment to say that on behalf of everybody at Reasons to Believe, we just want to let you know how much we appreciate each and every one of you. Thank you so much for your willingness to invest your time and energy and resources in partnering with us at Reasons to Believe. It means a lot to us to know that there are people that are willing to align with us in the mission that God has called us to, and to be active partners with us as we seek out to show people how science points to God and how science can be used as a bridge to the gospel. And in many respects, this presentation tonight is really addressing that particular issue. And the genesis of this talk goes back to a conference that I was invited to speak at for the Christian Scientific Society, where the whole theme of the conference was, can evidence actually lead people to faith in Christ? And, it's pretty commonplace, I think, for both people that are believers as well as non-believers to acknowledge that science and faith can be compatible with one another, that science and faith can exist in harmony with one another. And when people say that, oftentimes what they really mean is that science operates in one magisteria, to use the terminology of the late Stephen Jay Gould, and religion or faith operates in a different magisteria. And because these magisteria are distinct, of course, science and faith can harmonize with each other. Science and faith can actually be compatible. But what we're looking at this evening is actually going one step deeper in terms of that compatibility and that harmony, to actually argue that there are scientific insights that actually can inform our faith, and that science can actually provide evidence for belief in God. And now when you make that assertion, you are actually arguing that the two magisteria actually overlap, that they interconnect with each other, and that you are, in the minds of many people, dissolving the boundaries between science and faith, and that you are forcing science outside of its traditional boundaries, at least in modern times. And so this idea that science can provide a basis for faith is really, in many respects, a radical idea. And I think those of us who are part of Reasons to Believe are so accustomed to looking at scientific evidence for belief in God that we fail to realize how radical this idea actually is, and how uncomfortable it makes people feel to make this assertion, not only for non-believers, but also for even people within the church. both people that are of a scientific orientation and people who are also of a theological orientation. And so this is really, I think, in many respects, a very important question, and one that's really worth thinking through in some detail. And so that's what we're going to do tonight. Now, when you ask the question, can science provide evidence for faith or provide a basis for faith? This is really an umbrella question that sits on top of another series of questions, like, what do you mean exactly when you talk about science? What do you mean exactly when you talk about faith? If you're talking about faith in God, what is your concept of God? And is it possible to identify evidence for God? And if so, does that mean that this God has chosen to reveal himself to us? And how would that revelation look? And so these are all questions that are intertwined with this larger question that we are addressing, and we will indirectly touch on all of these sub questions, if you will. Now, when we talk about belief in God, of course, we need to be specific about what we mean when we talk about belief in God. And so we are, Christian theists here, or I think many of us are at least. And of course, as Christian theists, one of the most important aspects of how we understand the concept of God is that God is personal, and God is also triune, but that we believe that God is transcendent, meaning that, of course, that God exists outside and independent of his creation. We believe that God brought the world that we live in into existence, but that he is separate and distinct from the creation that he has made. And this idea of God's transcendence is not unique to Christianity, of course. This is a concept that Judaism and Islam also embrace as well. But what we see, for example, in Islam, is that the concept of God's transcendence is carried to an extreme. And now I'm not an Islamic scholar by any stretch of the imagination, but I did grow up in a home where my father was a devout Muslim. He was also a nuclear physicist, and had his own science faith struggles, I guess, and worked through the relationship between science and his faith as well. But again, growing up, the religion that I had exposure to primarily was Islam. And in fact, I even dabbled in Islam as a high school student. When I was about 16 or 17, I explored Islam in a serious way. I recited the Shahada, which is the declaration that Allah is the one true God and that Muhammad is his one true prophet. My father taught me how to pray. And so I would pray facing Mecca, and I would read from the Quran. So while I'm not an Islamic scholar, I do have some awareness of Islam. And in Islam, the idea of God's transcendence, again, is carried to an extreme. In many respects, God's transcendence is the dominant attribute of God that Muslims understand. And the way this cashes out is that God is unknowable. Because God is entirely and wholly transcendent, for a Muslim, God is inscrutable, he is unknowable. And this is an interesting idea because it means that God is hidden from us, according to Islamic theology. And what is interesting to me about this concept is that it actually sheds some light into a problem called the problem of the hiddenness of God. This today, for many philosophers, is considered to be the preeminent challenge to Christian theism. And it's related, interestingly enough, to the problem of evil, where many people would refer to God's hiddenness as the problem of divine evil, where the idea here is that if God exists and he wants to be in a relationship with the creatures that he has made, then he should fully reveal himself to us so that we can know him and that we can worship him. And the fact that God has remained hidden, they argue, it bespeaks of essentially the an unfairness on the part of God. How could God, in a sense, judge those people who reject belief in him if he indeed remains hidden from him, or sorry, remains hidden from us? This is the hiddenness of God problem. And so for many respects, it's considered to be, again, the problem of divine evil. But what we see in this concept of transcendence is, I think, a partial response to this very significant challenge. And that is to recognize that because God is transcendent, endemic in his transcendent nature is the fact that he's actually hidden from us. That God's hiddenness is endemic in saying that God is transcendent because he exists independent and outside of his creation. And so to say that God is hidden, again, is not something that God is willfully doing to frustrate belief in him, but rather is, again, endemic in his nature. But now, in terms of Islamic theology, all that can be known about God is what God wills. All we can know about Allah is what Allah wills, according to Muslims. And again, God is unknowable. And in fact, to make the assertion that you can somehow know God is considered to be a blasphemous statement, because what you are doing when you say that you can know God in some way, that you can understand God in some way, is to elevate yourself to God's level. You're saying that you are like God if you can know him, if you can understand him. And so this is considered to be a statement of blasphemy. But what we see in Christian theology, and again, this relates to the problem of the hiddenness of God, is that counterbalancing this idea of God's transcendence, of course, is the concept of God's eminence. That is, God is present and active within his creation. And this is not only the record of nature, but also as in each of us as individuals. And God is also active in human history. And it's because of God's eminence that God is now known to us, that we can apprehend God, at least in some capacity. And this leads to, of course, another important idea that flows out of God's eminence, and that is the idea of revelation. That yes, indeed, God is inherently hidden from us because of his transcendent nature, but he has chosen to reveal himself to us. And this revelation is intimately interconnected to the idea of God's eminence. And of course, Christian theologians would argue that this revelation is in two general categories. General revelation, which is a revelation that God has made available to all people at all times under all circumstances. And this includes God being revealed through the record of nature, God being revealed through his providential action in history, and then last but not least, God being revealed through human nature. Because we are image bearers, we are in effect, as we image God as image bearers, we are in effect revealing God in some respects. And of course, we also recognize that God has interacted with people at specific times in specific ways. And these interactions have been recorded in the Old and the New Testament and constitute special revelation, with the ultimate revelation of God being the person of Christ himself. And so we, as Christian theists, we recognize that the response to the problem of divine hiddenness is to, again, recognize that, again, endemic in God's transcendent nature is his hiddenness, but that God wants us to know him and has taken deliberate steps to reveal himself to us. And this includes the record of nature. And of course, if science is about the investigation of nature, then it follows that if God is revealed through nature, then science should be able to uncover evidence for God's fingerprints. And so flowing out of Christian theology is this expectation that there actually should be bona fide scientific evidence that points to the existence and the reality of a transcendent personal creator. This is, again, what we would expect as Christian theists. Now, I actually came to faith in Christ because of what I believe to be God's revelation that's evident in the record of nature. And I think many of you are familiar with my story of how I came to faith in Christ. But after finishing up high school, I ended up abandoning Islam. And there's a number of reasons why I gave up on Islam. And I'm not going to go into those details here this evening. But suffice it to say that I gave up on Islam, and as a result of that, settled into a position of agnosticism, where I wasn't really sure if God existed or not. And I honestly really had very little regard for that question. I was more interested in doing what I could to prepare myself to go to graduate school to pursue studies in biochemistry. And so as an undergraduate student, I embraced the evolutionary paradigm as the explanation for the origin and the design and the history of life. And like many people that I meet at that time, I reasoned that, look, if evolutionary mechanisms can explain everything in biology, then there really isn't a need for a creator whatsoever. A creator seems to be superfluous. And so that being the case, I felt very comfortable actually as an agnostic. So in graduate school, however, my perspective changed rather dramatically. As a graduate student, I was, of course, doing a deep dive into the study of biochemical systems. I was beginning to do my own research. I was reading the scientific literature in a systematic way for the first time, having conversations with faculty members and other graduate students. And so I was really beginning to appreciate at a fundamental level the nature of biochemical systems. And on top of that, I was also learning as a graduate student to begin to think independently as a scientist, to draw conclusions and to have my own convictions about what the evidence says, regardless of what other people say, regardless of what the consensus was. And it was out of those experiences that I began to, first of all, marvel at the elegance and the sophistication and the ingenuity of biochemical systems. And this led me to ask the question, how do we account for the origin of these systems? This is the origin of life problem. And I wanted explanations that went beyond just simply evolution did it, but rather I wanted to know the specific details. I was at that point where I craved that type of understanding. And so on top of everything that I had by way of responsibilities as a graduate student, I began to study the origin of life problem on the side. And very quickly reached the conviction that chemistry and physics could not on their own generate the types of systems that we see that define biochemical systems. And I concluded at that point that there had to be ultimately a mind that undergirded life itself. And of course, if there is a mind that's responsible for life, who is that creator, and how do I relate to that creator? And to me, the answer was in the person of Christ, and it was in the Christian faith. But the point here is that my journey to faith in Christ was actually predicated on seeing what I believe to be bona fide scientific evidence for a creator within the record of nature. And in my experience, I don't think is unique. I know a number of people who have come to faith in Christ, or whose faith in Christ is strengthened because of what they actually see as evidence for a creator within the record of nature. And so anecdotally, I think people's experiences highlight the fact that, again, there can be a scientific basis for faith. But if we are going to effectively engage people both inside and outside the church that are uncomfortable with this idea, particularly people outside the church that are scientifically oriented, we really need to go beyond just simply what I would call an intuition of design. And we actually need to produce a robust scientific framework that actually helps us to identify legitimate evidence, legitimate signatures within the creation that point to the work of a creator. And so that's what I'm going to focus my time on, is how can we develop a methodology that we can apply in an objective manner that can show scientifically evidence for God's fingerprints within creation. And I'm going to focus on biochemical systems because, of course, I'm a biochemist. But when you, again, bring up this idea that there can be scientific evidence for faith, for belief in God, many people, again, who are scientifically oriented would push back very strongly against this, because what you're doing is actually dissolving a very critical boundary that people have drawn around the scientific enterprise, a boundary that's been influenced by the ideas of methodological naturalism. Yet when people argue that scientific evidence in being deployed to make a case for a creator's existence is outside the domain of science, they really are operating, I think, in a disingenuous fashion. And I say that with all respect to people that again are influenced by methodological naturalism, because I don't think they intend to be disingenuous. But there are actually scientific disciplines that are predicated on are predicated on the ability of science to be able to detect the work of intelligent agency in features within nature. And two disciplines that are prominent towards this end would be SETI, the search for extraterrestrial intelligence, where SETI researchers claim that they can examine, for example, electromagnetic radiation, and from the signatures in that radiation determine if it is generated through natural processes or generated by the intentional or unintentional activity of intelligent agents, of intelligent alien civilizations. Or archaeology, in which archaeologists can pick up a rock and tell you if that rock has been shaped by natural processes or if it's been shaped by the work of intelligent agency, some type of hominin. And so there are methodologies and there are tools within the scientific toolkit that actually can be used to detect the work of intelligent agency. And we can apply that methodology to biochemical systems to make a case that a creator is responsible for the genesis of these systems and the design of these systems. And so what I want to do is examine a very interesting study in archaeology that helps us to identify, in a sense, the strategy that we need to use to build a scientific case for a creator. And the archaeological study I want to talk about has to do with something known as the Sorretti Mastodon site. This is actually a site discovered in San Diego County, just outside of the city of San Diego, in 1992. And Caltrans was doing some road work and unearthed Mastodon remains. And of course, this brought everything to a halt. And a team of archaeologists went in and began to study these remains. And very quickly, they recognized that the Mastodon remains were actually processed deliberately by humans. That the arrangement of bones and cobble associated with the remains looked as if the remains had been processed after the Mastodon had died by humans. But at that time, nobody could date the site. And so the assumption was that this processing were done by some of the very first modern humans that made their way into the Americas somewhere around 10 to 20,000 years ago. In 2017, the researchers published a date for the site. They were able to successfully age date the site. And it turns out that the site is age dates to 130,000 years. This is remarkable because nobody believes that modern humans were in the Americas until about 20,000 years ago. And so the explanation that was being presented by the team of researchers was that most likely Neanderthals migrated into the Americas well before modern humans made their way there. This is an incredibly radical idea. And in order to defend this idea, the researchers were very careful to establish that indeed the remains had actually been processed by the activity of, in this case, what they believed to be Neanderthals. And so the first thing that they did is they established the artificiality of the site. In other words, they tried to demonstrate that the remains in the cobble had a non-natural arrangement, an artificial arrangement. And so this was in the juxtaposition of the cobble and the bones, but it was also determined based on markings that were made on the bones as well as on the cobble, that looked as if the cobble was being used to strike the bones in order to extract marrow from the interior of the bones. They then looked to try to exclude any alternative explanation. Could the remains have been modified by the activity of scavengers? Or could there have been water flow through the archaeological site that caused the bones and the rock to be arranged in a way that appeared to be artificial? And they ruled out those explanations, leaving that the only viable explanation was that these remains were indeed deliberately processed. And then they went into the laboratory and they took the femur of a dead cow, the femur of an elephant, and that these are fresh femurs, and they struck those femurs with rocks that were similar to the rocks at the archaeological site, and they were able to show that they could produce the same type of striation patterns that they found on the bone and on the cobble. And so on the basis of these three criteria, they concluded that these remains must have been processed by humans, by Neanderthals. That is, intelligent agency was responsible for the features that they saw at that archaeological site. And so this now gives us a roadmap for how we could actually establish scientifically that biochemical systems are reflect the handiwork of a creator. And we would ask the question, do biochemical systems display artificiality? Can biochemical systems arise through natural processes, through some type of chemical evolution? And then what does it take to create biochemical systems in a laboratory setting? And so what I'm going to do is very quickly just step through each of these criteria. The first is the criteria of artificiality. And one of the things that we talk about in the book, The Cell's Design, is the idea that biochemical systems display the appearance of design, display the appearance of artificiality. And what we note in that book is that the defining features of biochemical systems are identical to the types of features that we would recognize as the work of human designers. In other words, when human beings design, when we create, when we invent systems or objects or devices, those things that we produce have certain telltale signs that reflect the work of intelligent agency, that reflect the work of human agents. And we see those same features as the defining or hallmark characteristics of biochemical systems. And therefore, this can be used as evidence that these systems, again, have the appearance of design, display an artificial nature to them. In fact, there are some biochemical systems in their totality that actually resemble human systems in their architecture and in their operation to really an eerie degree. And this type of similarity allows us to use the watchmaker argument presented by William Paley in the late 1700s as a way to formalize this appearance of design into an organized framework that again highlights the artificiality of these systems, where of course Paley argued that a watch must be the product of a watchmaker, and therefore life must be the product of a divine watchmaker. through analogical reasoning, where Paley noted that a watch has certain attributes that reflect the work of a mind, and that he then generalized those attributes and then used that as kind of a intelligent design pattern, if you will, and then compared the attributes of living system to the attributes of a watch, with a rock as being the control, where we know a rock is produced through natural process mechanisms. And Paley argued that living systems, that biological systems, have more in common with a watch than with a rock, and therefore we can conclude, because watch requires a watchmaker, life must require, again, a divine watchmaker. And so we can demonstrate again, this idea of biochemical systems having the appearance of design in kind of a watchmaker context by looking at, just as an illustration, a couple of biochemical systems that I believe are astounding in terms of their sophistication and in terms of their appearance of design. And one category of biochemical systems are what are known as molecular machines. These are biochemical systems that literally are operating like machines inside the cell. And the molecular machine that I want to focus on is a machine called ATP synthase, or F1F0 ATPase. This is a massive protein complex that is embedded in the inner membrane of mitochondria in eukaryotic cells and in the plasma membrane of bacteria and archaea. It is a protein complex that is practically ubiquitous within the living realm, and is arguably one of the most important enzyme complexes in nature, because this is the enzyme complex that generates molecules called ATP, which are energy molecules utilized by biochemical systems to power their operation. In the image here on the screen, it shows two depictions of ATP synthase. On the left, this is called a ribbon diagram, and on the right, what we see is a space-filling model. And this protein is made up of a large number of individual protein subunits that aggregate together to form this particular molecular architecture. And it turns out that F1, F0 ATPase is an electrically powered rotary motor that takes the flow of protons that move through the F0 channel embedded in the membrane, and it's the flow of protons, and the flow of a positive charge is a current, but it's the flow of these protons that energize the motor and drive the rotation of a rotor that rotates relative to the motor. And that rotor has a cam at a right angle that will interact with these turbine type structures, these blue and green ovals, and that causes a mechanical displacement, a conformational change that then is used to drive the production of ATP from ADP and inorganic phosphate. And then there is a stator that holds this motor together. This molecular motor operates at near 100% efficiency, by the way. And it's one thing to show you a picture of the motor, but it's another thing to see the motor in action. And so what I'm going to show you is a computer animation of how biochemists think this motor operates during the course of its catalytic cycle. So this is, again, a computer animation that is informed by hundreds and hundreds and hundreds of individual studies on ATP synthase, where all these studies are put together to produce this computer animation. So let's just go ahead and watch this enzyme in operation during its catalytic cycle.
These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of two separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin. And at the top, we have the F1 motor from factor 1.
And so it's remarkable to see the machine-like nature of this enzyme. And what's also instructive is to look at how people that are skeptics who are atheists respond to the remarkable nature of this particular enzyme complex. One of them is Nick Lane, who is a biochemist and an origin of life researcher, and he's written a wonderful book that I would commend to all of you called The Vital Question, about his ideas on how life originated and his insights from a bioenergetics standpoint, again, written from an evolutionary perspective and from the perspective of a non-theist. But this is what he says about ATP synthase. The most impressive nanomachine of them all, ATP synthase, is indeed a rotary motor in which the flow of protons turns a crankshaft, which in turn rotates a catalytic head. The protein works like a hydroelectric turbine. This is barely poetic license, but a precise description. Yet it is hard to convey the astonishing complexity of this protein motor. This is precision nanoengineering of the highest order, a magical device, and the more we learn about it, the more marvelous it becomes. Some see it as proof for the existence of God. I don't, but it undoubtedly is a wondrous machine. So again, just the remarkable nature of this complex is clearly evident even to non-believers. Another set of molecular motors that I want to talk about are the proteins that manipulate DNA during processes like DNA replication, transcription, and DNA repair. And these proteins collectively operate as an ensemble of Turing machines during, again, different DNA metabolic processes. And in fact, because they're operating collaboratively as Turing machines, in a sense, we could think about processes like DNA replication as, in effect, a computer operation. And just to dig a little bit deeper into this, we need to spend just a minute or two talking about what Turing machines are. Turing machines are these abstract machines that were the invention of British mathematician Alan Turing in the 1930s. And Turing is considered by many people to be the father of modern day computer science, where he laid down the theoretical framework for modern day computers. And his Turing machine is conceptually is a simple machine that consists of an input and output and a finite control. And so this is a cartoon of a Turing machine where you have the input being, in a sense, digital information, a string of 1 and 0s, or electrical pulses in the absence of electrical pulses. And the finite control is, again, a machine that will alter the input, producing an output. And it alters the input according to a prescribed set of rules, producing an output. And Turing's brilliance was realizing that the output of one Turing machine could become the input of another Turing machine. So even though individual Turing machines could perform limited transformations of the data, collaboratively, they could actually carry out complex computations. And so this is the basis for how computer systems operate. And it turns out that, again, when DNA undergo processes like, let's say, DNA replication that's shown in this diagram, it's essentially an ensemble of enzymes that are carrying out this process. But as they carry out the process, again, the individual enzymes are operating as finite controls, where the information in DNA, which is digital information, serves as the input and the output. And collaboratively, these Turing machines, though they are only able to form limited manipulations on the DNA, collaboratively can form a very complex metabolic operation, namely DNA replication, that again, in effect, is a computer operation. And this is, again, another video put together by biochemists that illustrate the process of DNA replication. And when you look at this video, think about this in terms of Turing machines, of the finite controls that are manipulating DNA, where the DNA, again, is the input and the output. So again, it's the machine-like nature is rather remarkable, where again, DNA is the input and the output, and the enzymes that manipulate DNA are finite controls. In fact, this similarity between DNA replication and computer operations is so stark that it actually has inspired a new area of DNA computing, sorry, a new area of nanotechnology called DNA computing. And this is a picture of Leonard Adleman, who's the father of DNA computing. And Adleman, again, realized by reading a molecular biology textbook that, again, what happens when DNA is manipulated in the cell is a computer operation. And currently people are looking to build computers out of DNA. And these are wet computers that are housed in little tiny Eppendorf test tubes. And they are more powerful than the most powerful silicon-based computer, in part because of the capacity to do massive parallel operations simultaneously. But this is what Adleman said in an interview about DNA computing. The most important thing about DNA computing is that it shows that DNA molecules can do what we normally think only computers can do. This implies that computer science and biology are closely related, that every living thing can be thought to be computing something, and that sometimes we can understand living things better by looking at them as computers. And so again, this is provocative, but when we think about DNA computing or molecular machines in the context of the watchmaker argument, I think it highlights the artificiality of biochemical systems. I think when we look at biochemical systems, this first criteria is easily met. And that leads us then to the second criteria, which is can chemical evolution explain the origin of biochemical systems? And the short answer to that question is no, they can't. Origin of life researchers today struggle to provide any kind of genuine explanation for how chemical evolutionary processes could generate life and hence biochemical systems. Paul Davies, in his book, The Fifth Miracle, said, when I set out to write this book, I was convinced that science was close to wrapping up the mystery of life's origin. Having spent a year or two researching the field, I am now of the opinion that there remains a huge gulf in our understanding. This gulf's not merely ignorance about certain technical details, it is a major conceptual lacuna. And just to support this statement by Paul Davies, very quickly, I just want to point out that every approach that origin of life researchers have taken to try to explain how life could emerge through chemical evolution has led to essentially a dead end. And there are three fundamentally different approaches that have been used: replicator first approaches, metabolism first approaches, and membrane first approaches. And all three approaches are riddled with significant problems. Replicator first scenarios, again, suffer from a number of problems. I'm not going to detail them here this evening, but if you go to books like Creating Life in the Lab or Origins of Life, you can see firsthand what some of those problems are. But the problems are so significant that Leslie Orgel at an Origin of Life conference made this statement that it would be a miracle if a strand of RNA ever appeared on the primitive Earth. His statement is in reference to replicator first scenarios, in which the RNA World Hypothesis is the preeminent replicator first approach to the origin of life. The same problem also is associated with metabolism, metabolism first scenarios also are confronted with intractable problems as well. Again, I'm not going to detail them here. But Orgel, before he died, wrote a number of review articles in which he was critical of metabolism first scenarios. And in one of them, he said, these scenarios require an appeal to magic, a series of remarkable coincidences, a near miracle. And then last but not least, a number of years ago, I wrote a critical review article with another chemist by the name of Jackie Thomas, where we critiqued membrane first scenarios, highlighting again what we saw to be some significant problems with membrane first scenarios. So the bottom line is that when it comes to the second criteria, I think it's safe to say that we really have no bona fide explanation for how evolutionary processes could generate the very first cells and hence biochemical systems. So we've satisfied two of the three criteria. And so now we move to the third criteria, and this has to do with what does it take to create life in a laboratory setting. And of course, this question is being explored actively by origin of life researchers who oftentimes go into the lab and they try to develop, in a sense, a pathway of chemical reactions, chemical transformations that can move from simple molecules to the very first protocellular entities. This is part of a research program known as prebiotic chemistry, where again, researchers are trying to recapitulate the different steps they think took place during the process of chemical evolution in the laboratory. And in many respects, you could argue that the last 70 years of work in prebiotic chemistry have been wildly successful. That origin of life researchers have identified a number of chemical routes that could produce virtually every one of the building blocks, that could generate biopolymers, and could even generate vesicle systems, protocellular systems. But on the other hand, you could argue that while these experiments have been successful on one hand, they have failed ultimately because they have not shown that the work in the laboratory could actually have any relevance to the conditions of the early Earth. That is, they failed to show geochemical relevance to the prebiotic reactions they have discovered in the laboratory. And what I mean by that is this, that it's one thing to go into a lab and to set up an experimental apparatus to establish the initial conditions of the reaction, to add the just right compounds in the just right order at the just right concentration, to have chemically pristine systems that exclude any kind of interfering materials, and then to run the reaction under highly controlled conditions where you're stopping the reaction at the just right point in time to preserve the integrity of the products that are produced. It's one thing to do that in a laboratory, but then to translate those laboratory conditions to the conditions of the early Earth becomes problematic, because these conditions in the laboratory are established by researchers, where the researchers have actually incorporated their role as the researcher, as the investigator, into the experimental design in ways that are unwitting, and yet you would not have that oversight or that control under the conditions of the early Earth. And so it's hard to argue that the laboratory conditions could ever, or that the laboratory work in prebiotic chemistry has any kind of relevance to the conditions of the early Earth. This is what Clemens Reichertz, an origin of life researcher, said recently in a commentary piece published in Nature Communications, because increasingly origin of life researchers are recognizing the problem of unwarranted researcher involvement. And this is what Reichert said: "It's not easy to see what replaced the flask, pipettes, and stir bars of a chemistry lab during prebiotic evolution, let alone the hands of the chemist who performed the manipulations. And yes, most of us are not comfortable with the idea of divine intervention." And let me just illustrate this by talking about a set of experiments that were initially done in the early into mid 1990s and actually continue to this very day, that many people take as support of the Rna World Hypothesis. And these experiments involve the assembly of RNA molecules on clay surfaces. And the idea here is that if you could actually assemble RNA molecules from RNA building blocks on clay surfaces in a laboratory, that means that maybe on the early Earth, clays that would have been present could serve as sites that could sequester RNA building blocks and then provide a protective environment that would prevent them from undergoing hydrolysis in an aqueous environment. And then also the clay could provide catalytic functional groups that could drive polymerization reactions. The problem is that these incredibly remarkable experiments done in the laboratory probably lack any kind of geochemical relevance and are characterized by researcher intervention, where the researcher's role has contributed to the success of the experiments. So, for example, in all of these experiments, researchers are very careful to exclude materials that would promote the hydrolytic breakdown of RNA chains once they formed, or would inhibit their polymerization. And yet many of these materials that have been excluded from the experimental design would have been present at very high levels under the conditions of the early Earth. They also stopped the polymerization reactions or the RNA polymers got too long, because they observed that once the polymers get above about 50 subunits in length, they become irreversibly adsorbed onto the clay surface. And this is about the size that you would need to begin to have RNA molecules that could function as catalytic agents, as ribozymes. And so the conundrum here is that if you let the chains get too long, they're never going to be available to form ribozymes. And then last but not least, in order to pull off these experiments, the researchers had to buy clay from a particular supplier. They had to treat the clay in a special way. And then they had to actually use chemically activated building blocks to ensure the polymerization reaction took place. Paul Davies said this: as far as biochemists can see, it is a long and difficult road to produce efficient RNA replicators from scratch. This conclusion has to be that without a trained organic chemist on hand to supervise, nature would be struggling to make RNA from a dilute soup under any plausible prebiotic conditions. Simon Conway Morris says this: many of the experiments designed to explain one or other step in the origin of life are either of tenuous relevance to any believable prebiotic setting, or involve an experimental rig in which the hand of the researcher becomes, for all intents and purposes, the hand of God. And so we see here that work in prebiotic chemistry, where we're trying to create protocells in the laboratory, actually indicate intelligent agency seems to be an indispensable component in the generation of building block materials and the assembly of those materials into functional biomolecules as critical steps in the origin of life process. But we also see that there's another area of work that also involves attempts to create life in the laboratory, and this is work in synthetic biology, where the goal in synthetic biology is to create artificial, non-natural organisms. And there are a number of different approaches that synthetic biologists are taking towards this end. And one approach involves the creation or the generation of protocells. And it's interesting because the work that synthetic biologists engage in as they try to create protocells is in effect the same type of activities that scientists working in prebiotic chemistry are engaged in. And in fact, today, synthetic biology and work in prebiotic chemistry are cross-informing one another. There's a lot of crosstalk between these two disciplines, where some of the best work in prebiotic chemistry is being done by synthetic biologists, and some of the most important insights that synthetic biologists are taking advantage of come from work in prebiotic chemistry. And so we would expect then that the work in synthetic biology should yield a similar type of conclusion as the work in prebiotic chemistry, where again, ironically, the work in prebiotic chemistry designed to try to demonstrate the viability of chemical evolution has actually shown the necessary role of intelligent agency. And so there was a study that I love to talk about towards this end, where a team of Japanese researchers created self-replicating protocells. And this work was published in 2015. So it is a bit dated in many respects. It's about 6 or 7 years old now. But yet this work, I think, represents kind of a quintessential study in synthetic biology and in the creation of protocells. In many respects, this work is really science at its very best. These Japanese researchers created protocells that could actually grow and replicate for multiple generations. and they were able to couple the replication of the protocells with the replication of DNA within the protocell's interior, and they actually devised a way to replenish the building block materials in the protocells after the cell division process took place. So they had a really sophisticated system that they created. And here's a cartoon showing an overview of the work that they did, where they created these vesicles that contained in the vesicle interior nucleotides, which are the building blocks of DNA, small pieces of double-stranded DNA, something known as a DNA primer, and then an enzyme called DNA polymerase. And in order to form the membranes, they used particular phospholipids that had the just right set of properties that would allow them to form stable vesicles that could again house these different biomolecules. They then heated up the vesicles very carefully, which would cause the DNA double helix to unwind. And after it unwound, this now exposed single-stranded DNA that then, through the use of the primer and the nucleotides and DNA polymerase, could assemble daughter DNA molecules, producing double-stranded DNA. Now, one of the components in those that were used to form the wall of these vesicles was a specially designed molecule that was designed and synthesized by the research team. And this molecule bears a positive charge. And so they adjusted the concentration of this particular component in the vesicle so that the interior walls of the vesicle would have a positive charge. This would allow the DNA double helix, which is negatively charged, to adhere to the vesicle wall. And this is how they were able to couple the growth and the division of the vesicle with DNA replication. They then designed and synthesized another type of membrane component that they could add to the vesicles after DNA replication took place. And this component incorporated it into the membrane, causing the membranes to swell. And eventually the vesicles got so large that they would become unstable and they would fissure into two daughter vesicles. They then added another type of vesicle that they created that had nucleotides and DNA primer materials incorporated into them, that they then and they designed the vesicles to have a negative charge so that they would through electrostatic interaction absorb to the daughter vesicles. Then by altering the pH of the solution dramatically through a pH jump, they were able to get the vesicles to fuse, and voila, they wound up with vesicles that were, again, the same type of vesicles that they started with. And they could repeat this process over and over and over again for multiple generations. So this is, again, a remarkable system, but it is a system that has been elegantly designed to carry out these processes that are reminiscent of the processes that living cells carry out. In order to do this, they had to choose the just right phospholipids for both the vesicles that would grow and replicate as well as the conveyor vesicles. They had to design two non-natural lipids and then synthesize them in the lab and very carefully adjust the composition of all the vesicle components in order to form stable vesicles that could then grow and replicate. They also had to very carefully control the solution conditions throughout the process, adjusting the temperature or the pH very carefully in order to, again, trigger the different steps in the growth and division process. And so this is what the researchers say in the conclusion of their paper. To achieve this goal, we selected well-defined suitable lipids and macromolecules, including newly designed ones, and constructed a giant vesicle-based model protocell that links self-replication of information molecules with the self-reproduction of a compartment. So in other words, they're utilizing design language when they talk about the work that they did. And you can see that if it wasn't for the role of these intelligent agents that developed this elaborate strategy that then executed this strategy in a very careful, precise way under controlled conditions in the laboratory, it would be impossible for this type of system to ever come together just simply through chemical and physical processes alone. And so again, this work demonstrates the central role intelligent agency must play in the creation of protocells. So the bottom line is this, that the three criteria that an archaeologist would use or a SETI researcher would use to establish evidence for the work of agency in nature can be applied to biochemical systems, showing that it looks as if systems are the product of an intelligent agent. And so not only, in a sense, could we argue that there is scientific evidence in biochemistry for a creator's fingerprints based on a kind of an intuition that we would have by looking at the nature of these systems, we can actually go one step further and construct a formal methodology that allows us to determine whether or not a feature in nature is indeed the work of agency. That this is established through rigorous scientific investigation. It is not, again, something that is a conclusion that would be drawn outside the boundaries of science, but a conclusion that we would draw within the boundaries of science. Now, where we move beyond the boundaries of science is to ask the question, who is this intelligent agent? But I would even argue that there are features within nature that begin to give us some insight into the capability and the motivation of this agent as well. That is, given the fact that the universe that we live in, the local universe that we're part of, has a beginning, suggests that there's a transcendent cause that brought the universe into existence. The fact that we see design in the fundamental constants of the universe and design in biological systems suggests a personality that's behind the universe itself. And so that is, even from science itself, we can begin to get some sense as to what is the capability and maybe what is even the motivation of this creator. Because you could argue that the fine-tuning of the constants of nature suggests a fitness for purpose, and that purpose seems to be in part the advent of sentient creatures like human beings. And so you know, the scientific data not only can establish, again, evidence for the work of agency, but can give us some information about who that intelligent agent might be and what that agent's capability might be and what that agent's motivation might be. And that then leads us to asking questions that are really theological in nature, namely, how do I relate to that creator, if at all? And this is where I think faith now becomes an important aspect of this conversation. So anyway, I'm going to go ahead and stop there and open up the floor for questions and for discussion.
Asking, have you heard of the Miller-Yuri experiment? And I know you have, Fuz, but maybe you could give us a little bit of insight into what the Miller-Yuri experiment did.
Yes, and as you said, Kirby, I've heard of the Miller-Urey experiment, and I've written about it in both Origins of Life as well as in Creating Life in the Lab. And you know, this is an experiment that is historically really rather significant, because that experiment really inaugurated origin of life research as a bona fide scientific discipline. And you know, the experiment was done in the 1950s by Stanley Miller, who was a graduate student of Harold Urey at the University of Chicago. And the experiment was done to validate the Oparin-Haldane hypothesis, advanced independently by the Russian biochemist Oparin and the geneticist from the UK, Haldane, who produced these stepwise mechanisms for how they thought the very first life forms could have emerged on Earth. And so because this was the first experimental validation of that idea, it really was a landmark in terms of establishing a roadmap for how the origin of life problem could be investigated. And the bottom line is that Miller showed that under what people thought to be the conditions of the early Earth, you could generate amino acids and other organic materials by passing electrical discharges through a simulated atmosphere of the Earth. The problem is that the conditions that Miller used in that experiment are not, as it turns out, to be the conditions that we now believe to exist on the early Earth. And when you actually use a more appropriate or more accurate conditions in the simulation, you wind up actually getting a null result. Nothing forms in that experiment. So most people today think that the experiment is scientifically irrelevant, but historically significant because of its importance in launching, again, the Origin of Life research program as a bona fide scientific discipline. So, in many respects, the problems that you see with the Miller-Urey experiment is essentially what I was pointing out when I was talking in general about prebiotic chemistry, is that it's one thing to go in a lab and to set up an experiment in which you can generate amino acids, but it's another thing for that experiment to have relevancy to the conditions of the early Earth. And even if the atmosphere that Miller used was the atmosphere that we would have found on the early Earth, the results are still irrelevant, because what Miller ended up showing in subsequent studies is how exacting the gas compositions have to be in the headspace in order for amino acids to form. That it's just not simply any random mixture of methane, ammonia, and hydrogen that will form amino acids. You have to have very precise ratios of those gases in order for amino acids to be produced. And if you deviate too much from that ratio, then you wind up getting nothing either, even under what are called reducing atmosphere conditions. So yeah, I've heard of the Miller-Urey experiment, and it's well worth, I think, if you're interested in this aspect of science, scientific apologetics, to be well versed in what the experiment is and how people view the experiment today, because it does show up in biology textbooks. And I do meet a lot of people who learn about this experiment, and on the basis of that single experiment are convinced that chemical evolution can explain life's origin. So because of its prominence, because it shows up in biology textbooks, and because people are familiar with it, even if they don't know much about science, it's well worth, again, being well versed in engaging that particular experiment.
Thanks, Fuz. Unfortunately, I think the person that asked that question may have left the meeting. Is John M. here? And if you are, would you please identify yourself? So I guess John M. has left. Anyway, I have a kind of a follow-up question to that, though, and that is that it seemed like to me that you implied that that researchers have actually created life in the lab, and it's just that if they go out into the field, they wouldn't be able to duplicate that in the field. So I don't think you meant that, but that's kind of the impression that I got.
Oh, okay. Yeah, I don't think, yeah, I didn't, if I gave that impression, I didn't mean to. And of course, part of the challenge is to be able to properly define what life is and what life isn't. And because nobody can truly define what life is, it's really a moving target in terms of whether or not a particular experiment has created a bona fide life form or not. But I actually am of the opinion that we will eventually create protocellular entities that we will recognize as living systems. I think it's just simply a matter of time. It may not be any time in in the next, decade or so, but I think eventually it will be something that researchers will achieve. The progress is pretty remarkable in my view towards that end.
This is a comment, but I'd like to have you comment on the comment. And Kent Gordon says, I think irreducible complexity is the best argument, especially for those not well versed in molecular science. So would you comment on that?
Yeah, this is a great comment because it really highlights, I think, the challenge that we all face as we are trying to engage people with scientific evidence for the Christian faith. Because what we want to do is make sure that we're able to present information that is accessible, particularly to people that are non-scientists. But the danger in doing that is that we can oversimplify things, and in doing so, actually undermine what we're trying to do, both directly and indirectly. Because if as we're trying to simplify our case and we present a simplified case, there are going to be people that will hear that case that are skeptics who are well versed in molecular science, molecular life sciences, who will simply walk away thinking there's no argument here at all. And the danger also is that if we present information that is accessible to lay people, they may turn around and then, understanding that argument, use it themselves and find that the argument doesn't go very far, particularly for people, again, that are well versed. And I've seen this time and time again where Christians, thinking they understand, who aren't, again, well versed in the sciences, who are well meaning, think they understand the scientific case against evolution, and then will go out and use arguments that they've heard and wind up just getting eviscerated by people that know something about the discipline. And sadly, irreducible complexity falls in that category. It's a very easy argument to understand, and there is some value in the argument, but in my experience, the argument plays very poorly among people that know anything about the molecular life sciences or about biology in general. And let me just illustrate the problem here. And as I do this, this is not meant to denigrate Michael Behe, who I have enormous amount of respect for, but the idea behind irreducible complexity is that it's defining a system that is composed of a number of parts that interact in a precise way to accomplish a particular purpose. And that if you remove one of the parts or even modify one of the parts, the system no longer has function. And so this kind of a system has to come together all at once. It has to be assembled simultaneously all at once. You can't assemble that type of system in a stepwise manner in an evolutionary context, because if the system is incomplete, it's not functional, and therefore it's of no use for the organism, and hence natural selection will never select that system. It'll never make it through the filter of natural selection. And so the idea is that irreducibly complex systems have to come together all at once. They can't be produced in an evolutionary framework in a stepwise process. And so biochemical systems are irreducibly complex systems. There's absolutely no question about it. And so Michael Behe's argument was, because these biochemical systems are irreducibly complex, they cannot be produced by evolution. Therefore, they must be the product of a designer. They must be designed. That, in effect, is how the argument goes. The problem is with that approach is that all somebody has to do is suggest a plausible way in which irreducibly complex systems could emerge in a stepwise manner, and they've essentially sidestepped the argument. And so evolutionary biologists do have a hypothetical way in which irreducibly complex systems can emerge. And it's through a stepwise process where there are intermediary systems, where those intermediary systems have functions that are distinct from the final system, and that through an evolutionary process, those systems or portions of those systems are co-opted and combined with other systems to create systems with new functions. And so it's through a process of co-option that a stepwise assembly of irreducibly complex systems can hypothetically take place. And so many people on that basis will reject the argument. I'm sympathetic to skeptics who reject the argument for that reason, because I think, again, the argument doesn't handle this mechanism of co-option very well. And if you notice that much of what Michael Behe has done subsequent to presenting this argument in his book, Darwin's Black Box, is essentially to debate people about whether or not examples that they've presented that they think illustrates the phenomena of co-option are legitimate or not. And so what's happening is there's this whole sub-debate going on between Michael Behe and his critics about whether or not the counterexample that they present to the evolutionary origin of irreducibly complex systems, again, are legitimate or not. So to me, I would actually prefer a very different approach than this particular approach, which is, in a sense, I would advocate the approach that I'm presenting tonight. And I actually think that this approach is something that takes a little bit more effort to present, but I think it can be presented in a way that is fairly accessible to lay people. I mean, to talk about how an archaeologist goes about detecting the work of agency is, I think, accessible to most people. And I think it's easy enough to make the points that I made tonight with less detail and more conceptually, and I think make a case that non-scientists could understand. So I'm not convinced that we have to go down the route of irreducible complexity. Now, if you like the argument, I'm not saying don't use it. I'm just saying that you should really be aware of the pitfalls of the argument, that you need to be aware of the pitfalls. And I've actually found that, and again, I don't want to say this in a way that throws Michael Behe under the bus, because I admire him greatly, but I found that engaging skeptics, if I actually agree with them that irreducible complexity isn't a good argument, it actually gains me a hearing with them. And I've had a number of instances where I've gotten pretty deep in the conversation with skeptics because I was willing to say that I agreed with them that this isn't the best argument.
We have a question from Ron Martin. He says, does the vesicle first account of abiogenesis start with protonoid spheres and assume that ports for admitting and expelling molecules came about?
The very earliest ideas, for the origin of life were in this category, where you kind of like a coacervate idea. And there's probably a few people that still advocate the role of coacervates in the formation of the very first compartments. But there's a number of origin of life researchers who point out that in the Murchison meteorite, you can recover lipidic materials that will assemble into vesicles in the laboratory under specific sets of conditions. And so they argue that that there were primitive vesicles made up of fatty acids and other type of lipidic materials that would have been present on the Earth that could have formed these compartments. And these compartments would have had a high degree of permeability, which would allow the flow of materials in and out of the compartments. So it's actually a bona fide membrane first scenario where the vesicles are made up of fatty acids and fatty alcohols and things like that.
Okay, Tom Kennedy has asked a question. I think that repeats some of what you've just said. Not this question, but a question that I asked earlier. I'm going to read his whole question, but I think that maybe we could focus on the second part, since I think you have answered the first part already. As you seem to have said that significant proto-life structures can be created in the lab setting, but this is not relevant to the conditions on prebiotic Earth. Dr. James Tour just posted a series of lectures where he seems to claim that little can be created even in a lab. Are you familiar with Dr. Tour's work, and does your understanding differ greatly from his?
Yeah, I am familiar with Dr. Tour, and I think many of the arguments that he would use, I would use as well. But to me, I guess I would disagree with him in the sense that very little has been done successfully in the laboratory by original life researchers or by synthetic biologists. Because when I go into the literature, I see significant steps that have been made towards creating protocellular systems that have remarkable properties, that I think original life researchers have produced a very impressive body of work, identifying in principle different chemical and physical processes that could contribute to the origin of life. And they've done really elegant work in terms of dissecting the mechanistic undergirding of these chemical and physical processes. To me, I think where the significant breakdown takes place isn't in the inability of researchers to do this, to identify these chemical processes in the lab, but rather it's how well do these processes translate to the conditions of the early Earth, where I think you could make a very robust argument that these systems again lack geochemical relevance. And the approach that I take has been an approach that I've tested with origin of life researchers. When I've engaged in conversations with origin of life researchers, if you acknowledge the work that they've done and the progress that they've made, but yet identify again this issue of geochemical relevance, almost to a person that they will agree with you. They will recognize. In fact, the quote that I put up from Clement Reichertz actually acknowledges, he's an original life researcher that acknowledges the problem and the fact that this is pushing you into theological territory. It's pushing you into the arena of intelligent design. If you notice that his parenthetical statement that we are not comfortable with the idea of divine intervention. And so to me, I think we want to be thoughtful about being fair to people that are working in these areas, and to acknowledge where they've actually done good work and where they have actually satisfied the criteria they need to establish a chemical evolutionary origin of life, which is do the processes exist that would be needed for the origin of life, and then B, can they operate under the conditions of the Earth? And I think the first criteria has been well established, and again, I think we want to make sure that we're giving due where it is due, the proper due where it is due to people that are working in these areas. And in my experience, when you're willing to do that and you actually are engaging the issues that they see, that the issues that keep them awake at night, they tend to be much more open to having the conversations that are pushing them towards the intelligent design arena. Let me tell you a story, and I tell you this story actually with a heavy heart. And a number of years ago, I appeared on the Unbelievable podcast with Justin Brierley, and I was debating. I'm drawing a blank on his name now. I was debating an editor, one of the editors for Nature magazine. Adam Rutherford is his name, and he had just written a book called Creation, and it was essentially a secular version of my book Creating Life in the Lab. And so Adam Rutherford and I did 2 programs that were an hour long, where we had a discussion about the origin of life and a discussion about work in synthetic biology. And off the air, Adam Rutherford said to me, "You're the first ID person I've engaged or have read or have engaged or interacted with who actually understands the science." And that really made me very sad, because it means that the perception that many people have in the scientific community is that the Christian apologists that are engaged in this area haven't properly represented the science. And that makes me really sad to hear that. So, I think we wanted to be forthright about where progress actually has been made. We want to be honest and we want to be charitable. We want to give the benefit of the doubt to people that hold differing perspectives than we do. And what I find too is within synthetic biology, I mean, the amount of effort it takes to create these protocellular systems is legion. I mean, these are the best minds in the world that have developed these very impressive, ingenious, strategic approaches to creating these protocells, and then have executed them with, again, excellence that requires the greatest of precision in the laboratory. And so these are incredible scientific accomplishments. And to me, I think by paying a compliment to the researchers that have done this work, we have actually contributed to the case that we're making by acknowledging that this is impressive work, that this is a really significant accomplishment, actually feeds into the case that we're making for intelligent agency. And so we can simultaneously compliment the people that have done the work, and by doing so, highlight their ingenuity, highlight the brilliance of the workers that have done the work. And so my concern, with all due respect to Jim Torr, who I admire greatly, by going to such an extreme where we are hypercritical of the work that's being done, we actually may be doing a damage to the work or to our case. That's my point. We just want to be careful about how far we extend the criticism and ask ourselves, are we truly being fair when we level this criticism?
Okay, I'm going to try to even the questions out so that one person doesn't get all the questions. Charles Franks says, do you still think that the DNA code is the best biochemical evidence for the existence of a creator's existence?
I would say when it comes to biochemistry, the one piece of evidence that I talked about tonight, the fact that DNA metabolic processes are in effect computer operations, to me is mind-boggling, and the implications are really, again, mind-blowing. I would also say it's the design of the genetic code, which is not the same thing as the DNA code. The genetic code is actually the set of rules that the cell uses to translate the information in DNA to the information in proteins. And when you look at the nature of the genetic code, it's absolutely astounding, the degree of optimization that you see in the genetic code. And it's optimized for a number of different factors. In October, I have a new book coming out called Fit for a Purpose, where I present a case for the biochemical anthropic principle and the idea that biochemical systems are fine-tuned, and they're precisely fine-tuned for life to be possible. In other words, that biochemical systems have to be the way that they are. If they're any other way, life could not exist. And what's really eerie is that it looks as if these biochemical systems are fundamentally prescribed by the very structure of the universe itself, by the fundamental laws of nature. And that even includes the optimization that we see in the genetic code, that it's fundamentally dictated by what are called the wobble interactions that take place during the process of translation at the ribosome, and that's highly technical. But anyway, so yes, the short answer to that is I think the DNA is a great system to focus on. I would again caution against using the arguments that I see a lot of apologists use about the improbability of generating biochemical information, because the more that I've studied that particular claim, the less compelling I find it. In other words, I don't think the idea of information the idea that I don't think the probability argument is a sound argument. It doesn't take into account certain things that we know to be true about biochemical systems, that if you took those into account, it would dramatically undermine the argument. So I would prefer making kind of a watchmaker argument, where we talk about the similarity between the design of these systems and in human designs. Now, when it comes to the genetic code, however, that is a place where I think we can show rigorously that the likelihood of evolution generating the genetic code that we see in nature is extremely remote, if not impossible. So that is a place where I think you can actually bring to bear a direct challenge to evolutionary explanations.
Okay, I've got several questions from Ron Martin. One of them I think has a very quick answer, and I'm going to try to answer it myself. If I'm wrong, somebody else, or you can correct me, Fuz. But he said, did Miller carry out Yuri's experiment in 1965, the 50s? He carried it out in 1953. I think that's the answer.
Yep. The same year that the structure of the DNA molecule was published is the same year that the Miller-Urey experiment was published.
And the second question that he asks is, does science get the nature of conditions on the early Earth by examining the state of planets in our solar system?
Could you repeat that again?
Does science get the nature of conditions on the early Earth by examining the state of planets in our solar system?
I think originally that's how the conditions of the early Earth were determined when Miller did his experiment in the 1950s. Harold Urey was interested in the atmospheric composition of planets, and if you look at, for example, Jupiter or Saturn, the atmosphere is made up of hydrogen and I think methane and ammonia, and so the assumption was that was also the conditions of the early Earth. And it very well may be that at the very early stages of the Earth, those are the gases that were present, but we now know that Earth doesn't have the gravitational pull to retain hydrogen in the atmosphere, and that ammonia and methane would undergo essentially decomposition reactions to produce nitrogen and carbon dioxide and water. And so many people believe that primary atmosphere would have been lost to those mechanisms, as well as to the impact events that were taking place, stripping the Earth of its original atmosphere. And so the atmosphere that would have been established on the Earth, that would have been available to the original life, would have been a secondary atmosphere. But people also gain clues from the geochemical composition of the oldest rocks on the Earth. They look for certain types of distributions of certain elements and other materials that would reflect whether or not the atmosphere was reducing or oxidizing. They try to estimate the number of the CO2 levels and things like that from carbonates in the rocks and things like that. So it's a number of pieces of information that are basically used as clues to try to reconstruct the conditions of the early Earth. So it's not just simply what we observe in our solar system, but it's other clues that are brought to bear. A lot of modeling studies are done.
Okay, and I'm going to ask one for myself. Fuz, what are some of the best arguments you've heard in support of common descent? And what are the best responses to these arguments?
Okay, to me, if there is a strong case that could be made for common descent, it would be basically the shared DNA sequences that we see in humans and the great apes, and particularly those sequences that presumably are non-functional. If you see shared non-functional sequences, and also the fact that you can build evolutionary trees where you see kind of a nested hierarchy with regard to these sequence properties. So I think that would be the best evidence for common descent. And to me, I don't know that you could necessarily refute common descent, but I think what you could argue is that there is a viable alternative, which would be, shared design. And that would be predicated on showing that A, the sequences that are shared are actually functional, B, that there is a reason why those sequences, there's a reason why those sequences look the way they look. So it's not just simply establishing function, but explaining, again, why the sequences are the way that they are. And then lastly, it would be, I think, making a case that the nested hierarchies are not as robust as you would necessarily think. And for example, when you compare the human genome, the chimp genome, and the gorilla genome, and the orangutan genome, with most of the sequences, you would get an evolutionary tree that would show the orangutan lineage broke off of first, and then the second lineage broke off would be that of the gorillas. And then finally, there's a bifurcation that leads to humans and then chimps and bonobos. But depending on the region of the genome you use, you actually get different trees. This is kind of one of these hidden secrets in molecular anthropology, that there's about a third of the sequences in the human genome that actually more closely align with the gorilla genome than with the chimp genome. And you also see the same thing with the orangutan genome, that there's a significant percentage of orangutan sequences that more closely align with human than they do with gorilla or chimp sequences, which means that if you use those sequences, you would actually get a very different evolutionary tree. And so this, I think, suggests I think undermines this idea that there's a nested hierarchy in these sequences. Yes, indeed it is, but that hierarchy depends upon the region of the genome that you use to build the trees.
I think I'm just going to limit us to one more question. And Ron Martin has asked it. This is a great follow-on question to what we've just been talking about. Do humans and chimps have non-functioning, that is, junk sequences in our DNA? And I'd extend that to orangutans and gorillas, etc.
I think that right now is a legitimate, outstanding question. Are there truly junk DNA sequences in the human genome or the genome of other organisms? And one of the radical findings of the ENCODE project in the phase 3, when phase 2 was reported, is that arguably about 80% of the human genome appears to be functional, as opposed to a original tabulation of only about 5% appears to be functional. And phase 4 of the project has been, phase 3 and phase 4 have just been published within the last year. And they don't give a quantitative assessment, but what they have done is identified even more functionality, where there are sequences that may be only functional at certain stages in embryonic development, but they're absolutely critical for embryonic development. So when you take into account not only the duration of the cell cycle, but also the developmental trajectory, there are probably almost every sequence in the human genome displays function. And I'm actually working right now on a series of blog articles on the functionality of endogenous retroviruses. And I probably will have a series of 4 or 5 articles over the next several months that I'll publish on my Cells Design blog. I published the first of those just a couple of weeks ago. But we've done a lot of work on pseudogenes, both in Who is Adam and as well as in thinking about evolution. There's a discussion of pseudogenes, but we've not done a lot of work with other areas. And so the next area that I think I need to focus on are endogenous retroviruses. And we're beginning to recognize that endogenous retroviruses actually serve an antiviral role. They actually are part of the innate immune system, and their ability to function in providing innate immunity against viral invasions is predicated on their similarity to retroviral sequences. And so there's a number of mechanisms by which they exert this activity. And I'm about to write up a blog article about endogenous retroviruses. Actually, these sequences are actually expressed at very high levels during the early stages of embryonic development. And the thought is that there's two things going on. One is that the transcripts from the endogenous retroviruses are actually mediating gene regulation that's critical for embryonic development in the early stages, and simultaneously are functioning in an antiretroviral sense, where they actually are protecting the embryo from retroviral invasions at the very earliest stages of embryonic development. And what ends up happening is the endogenous retroviral transcript actually competitively inhibits any kind of viral assembly in the cell. And the proteins that are translated from those transcripts are non-functional viral proteins that would form like the virus capsid and things like that, but they're non-functional. They won't lead to the functional assembly of a virus, but they will frustrate any kind of assembly process that's underway through competitive inhibition. So that's one mechanism that's been identified, and there's other mechanisms as well that have been identified. So it looks like, even the areas of the genome that people would have traditionally thought there's no way these things have any kind of functionality, we're seeing function for them.
I said that was going to be the last question, but I see that there's two more questions for an area that we haven't really touched on yet, and that's commenting on Stephen Meyer. And both of these are very similar questions, and I think you could maybe, if you could take one more question. Is that all right?
Sure.
Okay.
Yeah, and this isn't not meant as an attack against Stephen Meyer in his latest book or anything like this, because I see there's a lot of people that use the information argument or use probabilities with us in association with information to make an argument against evolution. And here's the problem, and there's a number of problems. Number one is what you're assuming when you do. Here's how the argument goes. Let's take a protein that's, I don't know, let's say 100 amino acids in length. And the probability of getting a precise amino acid sequence that's 100 amino acids in length would be 20 to the 100th power, right? Because there's 20 different amino acids that could be used at each position. There's 100 position, so it's 20 to the 100th power. That's the probability, and that's just an astronomical likelihood. It's a super astronomical improbability of hitting upon that precise amino acid sequence. The first thing that you're assuming, however, is that the amino acids are actually combining randomly. And there's a number of studies that suggest that that when you start combining amino acids in solution like that, it's not a random assembly, that there are actually selection effects. And in fact, original life researchers agree that it's not the random assembly that leads to information, but it's actually chemical selection. Now, you could argue that there's not a mechanism available through chemical selection that would give you the selectivity that you would need to create, again, functional proteins. But nevertheless, to argue that it's random assembly really doesn't properly represent what original life researchers are saying. So first of all, that's one problem that I see. We want to make sure we're not creating straw man arguments. The second problem is that we know by surveying proteins from the same protein that's found in a number of different organisms, that there are positions in that protein that can be varied rather freely, and others that are what are called conserved positions, where they have to be a particular amino acid. And so you have at a number of positions what's called functional equivalency, where there's more than one amino acid that could actually be used. And so when you start to incorporate functional equivalency, even if you are looking at random assembly, the probability plummets dramatically just by incorporating functional equivalency. Now, here's another problem, is that proteins are three-dimensional structures. And so what happens is many times that if you replace one amino acid in the protein with another amino acid, what was once an invariant position now becomes variable, and what were variable positions become invariant. And so it becomes this very complex computation when you start taking into account functional equivalency. It's not a linear problem. It's a multidimensional problem where you have to set up a matrix in order to do this, and nobody understands what the relationship between amino acid sequence and protein structure and function to be able to set up those matrices. So when you make when you're doing these probability calculations and you're making claims, you're claiming that you have more insight into protein structure than all the biochemists in the world have, right? So that's another problem that I have with that argument. Another issue has to do with ignoring physical chemical constraints. Just let me illustrate what's going on here. Let's say you took a protein that's 150 amino acids in length. Again, it would be 20 to the 150th power of primary amino acid sequences that would be possible. But each sequence has 10 to the 68 ways that it can fold, which means that this protein space that you're dealing with in these probability calculations is 20 to the 150th power, that quantity to the 68th power. Huge, huge, vast protein space. This is with three-dimensional structures, because this is what's critical for protein function. It's not the primary sequence. It turns out that there's only about 1 to 4,000 different ways that protein chains actually fold in nature. So even though you have this enormous protein space, there's only a small segment that actually can be occupied by stable folded proteins. There's only about, again, 1,000, anywhere from 1,000 to 5,000. There's only 10 to the third possible ways protein chains can fold. And we understand why that is. It's because of physical chemical constraints. The chains can't fold in any possible way they want because of those influences. And it turns out that those folds actually are highly promiscuous, meaning that they have the capability of carrying out a wide range of different types of functions. And so from those folds, you get the total functionality that you need to support biochemical systems. And so when you start factoring in physical chemical constraints, what you recognize is that thinking about protein assembly as a random process isn't the right way to think about it. It's these folds are actually prescribed within the laws of nature. In other words, when you're generating amino acid sequences, you are going to be generating proteins that will fold into potentially, into the high probability is they're going to fold into potentially functional products. Now, would that particular fold have the precise function that you're searching? No, but they would have a biochemically relevant function. And so this is all well established in the literature in protein science. And in fact, I have a whole chapter devoted to this in the book Fit for a Purpose, that again will be out in October. But the net effect is that it just simply decimates the probability calculations when you actually take into account those features. This idea of probability, just absolutely unravels. That argument unravels. But what replaces it is, I think, a more fundamentally sound design argument, because what you're actually saying is that the proteins that we see in biochemistry are not the product of historical contingency, but are fundamentally dictated by the laws of nature. And these are exactly the laws of nature produce proteins that have the exact necessary properties that are needed for life to be possible. And that if the laws of nature were any different, even subtly different, you would wind up producing proteins that would be unsuitable for life. So it's essentially an anthropic principle argument that you can make in exchange for this probability argument. But again, this is the type of argument that you present to somebody that has training in the molecular life sciences, and they will view you as somebody that doesn't know what they're talking about. And so it's a very persuasive argument for people that don't understand biochemistry. And for a layperson, it seems to be just this open and closed case for design and the impossibility of the evolution of life. It seems to be such a powerful case. And I see so many apologists using the argument, and it just isn't a good argument because it doesn't take into account what we truly know to be true about, again, biochemical systems.
Fuz, I think we've taken enough of your time, and I really appreciate your agreeing to come here and speak to us on this challenging subject.